Hello and welcome to the second of our tutorials in the Cardiovascular Anatomy series, and in this tutorial we're going to be looking at the anatomy of the heart and the pericardium and some of the disease processes that can affect these anatomical regions. So to start with, we're going to look at the embryology of the heart, and this will be quite a brief introduction into the embryology. It's by no means comprehensive. Do recommend you go away and have a look at that. And then we're going to look at the cardiac tissue structure, so we'll look at some of the cardiac embryology, but not too much to get too boring. The architecture of the atrium and the ventricles will then be looked at. We'll look at the architecture of the heart chambers and the various features that can help you to identify them, then look at the anatomy of the heart valves and their function, and then finally the pericardium and various different disease processes that can affect these different regions that we're going to look at today. Now, this is a very clever research study that was done in America, where they attempted to perform some MRI visualizations of human embryos, and what they've done is superimposed various different images on top of one another. They've taken the MRI images from various different angles, and then they've improved the resolution so you can see the embryos in isolation in a fair amount of detail. And what's quite remarkable is that they've taken the images of the lungs and the heart and they've isolated them in various different clever imaging methods, and so we can see the heart and the lung buds, and we can see how they're developing here. And this is a fairly well developed embryo. I can't remember the exact number of days, but they've superimposed the images of the heart and lungs here. So the heart is functional in the embryo. It forms a very important part of the developing embryo, but the fetal circulation is very different from the human circulation, because before you're born you spend a significant amount of time being a fish, and while you're a fish you don't need your lungs, and so the heart has to adapt to that. It doesn't want to waste blood in the lungs, so it has to develop various mechanisms to get blood away from the lungs, because the lungs are flat before we're born, and then when we are born it needs to quickly get rid of those mechanisms so that we can breathe in air. So we're now going to have a look at how the heart develops. The heart develops from that mesodermal tissue layer. So the initial embryonic trigeminal disc is made up of three tissue layers: ectoderm, from which you get your skin and your nervous tissues from; the mesoderm, where connective tissues and the cardiovascular tissues are going to come from; and then endoderm, which is responsible for forming organs such as those that are in the GI tract. We see something develop fairly early on the heart bud. We start to see the development of this tube. This is going to be the primitive heart. This snail, slug-like being here is going to be the primitive heart. Now, this tube is hollow. It's split into different portions that will eventually become the different regions of the heart. We start with this hollow tube split into the bulbous cordis, the primitive ventricle, the primitive atrium, and the sinus venosus. Ignore the vitelline veins for now. We'll look at those in a later tutorial. What then happens is that tube undergoes a coiling process. It twists round, and this is important in embryology in general. You'll see when you look at the embryology of the gastrointestinal tract that twisting, coiling and folding of various tissues is very important in orientation of structures. So this tube folds around, and this is when one of the first abnormalities can take place. You may have heard of a condition that's known as dextrocardia, where the heart sits facing towards the right rather than towards the left. In other words, it's the wrong way round. And this is sometimes associated with another condition known as situs inversus, where all of the organs are actually the wrong way round. And that's to do with very different molecular signals that determine the axis of the embryo not quite functioning correctly or functioning the wrong way around. And if that coiling process takes place the wrong way, then obviously the heart's going to be in the opposite direction. But what essentially happens in either case, really, is that the ventricle and the bulbous cordis are pulled anteriorly, and the atrium and the sinus venosus are going to be pulled posteriorly. Now, notice that we're saying ventricle and atrium. These are not divided yet, okay? We've just got one hollow space, one hollow cavity. So that coiling process effectively is starting to squash together what will become the heart. The truncus arteriosus at the top there is a new bit that we've not quite encountered. This is what's eventually going to form the aorta and the pulmonic arch, or the pulmonary artery. Now, the chambers themselves have to separate, and what we're looking at here is a schematic through the very primitive heart. So this square is as if we're looking at an empty, hollow heart chamber. So how do we get a hollow tube that looks something like this into 4 separate chambers? Well, what it depends on is ingrowths from various walls of those chambers. And firstly, we have these develop, what are called endocardial cushions. So from either side they grow inwards, and from the back they grow forwards, and they meet in the midline. But there is a space kept on either side that will eventually become the atrioventricular orifice. And this is on the right side, and the left side there's a slight little gap. These will eventually form the valve structures. We'll be able to see the atrioventricular valves in this region. So already we've split it into top and bottom parts. What then happens is from the top of the primitive atrium we start to get a downward growth that's called the septum primum. Now what's different about the septum primum is it doesn't completely form. There is a hole in the septum primum. It comes down to meet the endocardial cushion, so it does fuse with the endocardial cushions here, and it divides the atrium, the primitive atrium, into 2. So that's the septum primum. But you'll notice there's a hole in the middle, and that will become evident shortly. What then grows on the side of that is another membrane. This is quite thin membrane. On this schematic they're quite thick, but they're actually quite thin membranes. And there's another downward growth, the septum secundum. Now what that has formed is a valve-like structure. And this is what forms what we call the foramen ovale. And at the bottom we get the IV septum beginning to form. There's an upward growth in the middle of the ventricle, and this begins to split the ventricles into right and left portions. So this is all to do with ingrowths, and it can be a bit complicated this to start with. So let's go back to the beginning. So the first thing that happens is we get the ingrowths of the endocardial cushions. That's splitting the atrium and the ventricle into 2. We then get a downward growth in the atrium, forming the septum primum. To the right of that, we get the septum secundum, which covers that hole there, but leaves us tiny space, and then we get the IV septum forming. What eventually happens is when the truncus arteriosus gets involved and the vessels form, it brings a downward growth in, which shuts off that little hole in the IV septum. So now the ventricles are completely separated. To go back to that foramen ovale then, well, what's it important for? Well, it does this. The blood, instead of going down into the ventricle, goes straight into the left atrium to go around the circulation. And this alludes to what I was talking about at the beginning. The blood serves no use in going to the lungs. The lungs do not function in the embryo, in the developing fetus. They don't function. We're in an aquatic environment and they have no reason to function. And so what happens is blood is shunted. It's shifted from the right hand to the left hand side of the heart straight away through the foramen ovale. Now when the newborn appears, when they're born, when they're exposed to air, the lungs begin to inflate, and this sends various signals so that that septum secundum shuts down onto the septum primum. They fuse together and we get a remnant known as the fossa ovalis, which we'll talk about shortly. That was a very brief introduction to cardiac embryology. Please read the textbook. So the heart is a muscular pump. It's made up of muscle. It's a form of striated muscle because it's made up of sarcomeres, actin and myosin structures arranged in filamentous organization within the cardiac, what we call myofibrils or myocytes, the muscle cells. And they are interconnected, which means that they can contract as a sheet. Now, important features are the fact that they usually contain 1 or 2 nuclei. They are striated because they have sarcomeres in them. They branch so that they all connect together, and then finally we have these structures known as intercalated discs, and that's what this image is highlighting here. That sort of light structure in the middle there is an intercalated disc, and what that is a series of specialized proteins that allow the electrical signal, the action potential, to transport across this region here straight to the next fiber. So again, you get coordinated muscle contraction. So the structural organisation of the heart then, we have various different tissue layers. The very inside of the heart is on the left hand side, and this is known as the endocardium. Now this is made up of, it's a sort of squamous epithelium. It's a single layer of cells that lines the very inner chambers of the heart, serves a variety of different functions. The endocardium layer is like the endothelial layer of blood vessels. It detects changes in the blood. It also forms part of the heart's endocrine function, which is secretion of factors such as atrial natriuretic peptide. So the endocardium is a very important chemical controller as well. The most important tissue layer, though, is the next one, which is the myocardium. This is the muscular layer that's responsible for generating that forceful contraction that will expel blood into the systemic or pulmonary circulation. We then have the pericardium, which we will come on to in a bit more detail later, but essentially we have parietal pericardium, visceral pericardium that make up the serous pericardium, and then of course we have a pericardial cavity, and outside of that we have fibrous pericardium. So this is very essential to understanding cardiac function. So let's look at the chambers of the heart. The atria essentially have a smooth posterior surface, which is derived from that sinus venosus. It's actually, posh name is the sinus venarum. We then have a roughened anterior surface that's made of what we call pectinate muscle, which is derived from the primitive atrium. These two in the right atrium are delineated by a structure known as the crista terminalis. This is in the right atrium. And then we have a atrial septum separation that essentially splits off the two atrium. And you can get defects in that if those endocardial cushions don't fuse with the various different septums, then you can end up with an atrial septal defect. So this is looking at a very nice illustration of the right atrium. First thing that we can see obviously is a superior vena cava. That's being met by the azygous vein, which is draining from the hemiazygous system, from the azygous system of veins. We then have this ridge. This ridge is known as the crista terminalis. And in the right atrium, this delineates where the atrium is rough by the pectinate muscles, which are shown by these grooves here. And then we have this sharp bridge, the crista terminalis, and behind it we have a smooth surface, which is that sinus venarum. You can then see an opening down here. This is an opening from the coronary sinus. Now this is the main vein that drains the myocardium, but we will discuss that in the next tutorial where we talk about blood supply and nerves and innervation of the heart. We then have this, the embryological remnant that I discussed about the foramen ovale, known as the fossa ovalis. The fossa ovalis is a small depression, and if you were to hold the heart up to the light with the left atrium dissected, you would be able to see light passing through this. It's relatively thin. This is where the foramen ovale would have been in the embryo. The left atrium is very much the same. It has a rough anterior portion, which is comprised of pectinate muscles, and then posteriorly we have the sinus venarum, which is very smooth. You can sometimes see this depression, not always, which is the other side of the foramen ovale, and then we have 4 openings. Only 2 are shown here because of the way that this image is presented, but we would have 4 openings for the pulmonary veins that are carrying oxygenated blood back to the left atrium to go down into the ventricle. So it's largely the same as the right atrium. We don't really have that crista terminalis there. Instead, we've just got quite a sharp division between smooth and roughened anterior portions. Features of the ventricles. They have, in general, a greater amount of myocardium than the atria do. Basic stuff, A-level stuff. One of those facts that you remember to keep examiners happy is the fact that the myocardium is thicker in the ventricles because it has to pump it around the systemic circulation, whereas the atria have only got to pump blood a short distance. So it's only got to generate a small amount of pressure, whereas the ventricles have to generate a much greater amount of pressure. The left myocardium is significantly greater because it's got to pump blood around the body. It has trabeculated borders, what we call the trabeculae carni, and these are roughened muscles, trabeculated muscles, that essentially show the rough edges of the myocardium. We then have these features, papillary muscles, that are attached to these structures, the chordae tendineae of the valves, which maintain their patency, and we will come back to those again shortly. Features of the right ventricle, we have the pulmonary artery. Obviously the right ventricle is going to pump blood up to the lungs, and so this is known as the pulmonary trunk. Leading up to the pulmonary artery, pulmonary trunks, pulmonic arch, pulmonary trunk, etc. And this region here is smooth. The rest of the ventricle is rough, but this region here is smooth, and it's known as the infundibulum of the right ventricle. We then have that trabeculated surface, all rough, and then we have a particularly large band known as the moderator band, which sits in the corner. And it's probably in this diagram being shown by this region here. The moderator band is a part of the electrical conducting system that will be covered in the next tutorial, but the moderator band is important in propagating the electrical signals from up here to down here, etc. The left ventricle, as well as having a much thicker myocardium, has got a smooth surface again leading up to the aorta, and this is the infundibulum. So remember, you've got these rough and trabeculated borders and then a smooth area up here. The other important way that you can discriminate between these two ventricles in terms of spot tests, etc., is the number of papillary muscles. You get 2 in the left, 3 on the right. The valves. The valves sit either between the atria and the ventricles, the atrioventricular valves, or they sit between the ventricles and the great vessels, the aorta or the pulmonic arch. Now, one of the things that we come across a fair amount is how these valves work, and valve disease is quite common. The reason that valves are there is to ensure unidirectional flow, so that blood flows in one direction. And it's also there to make sure that blood flows smoothly, and if it doesn't, then we get what we call turbulent blood flow, and it produces what we call a heart murmur. The valves are responsible for producing the heart sound, the lup-dup, 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 that we listen for when we auscultate with the stethoscope. And the lup sound is the closing of the atrioventricular valves, and the dup sound is the closing of the semilunar valves. So you get a lup-dup, lup-dup, lup-dup. And if you don't get that, if you get an additional sound or you don't get that, etc., it can be indicative of cardiac pathology. So what do the atrioventricular valves do? Well, they sit between the atria and the ventricles, hence their name. They're pieces of very thin connective tissue, and they're anchored to what we call the papillary muscles that are around in the ventricle, and they are so by the chordae tendineae. They open in response to rising pressure in the atrium. So when the atria undergo systole, in atrial systole, pressure is generated much higher than the ventricles, and the valves are forced open. And again, as I said, they're responsible for the first heart sound. So you get first the tricuspid valve, which has 3 cusps, then the bicuspid or mitral valve, because it looks like a bishop's mitre, on the left side. I always remember tri before you buy, tricuspid before bicuspid, etc. Those chordae tendineae are responsible for holding the valves patent. So when the ventricles undergo systole, those papillary muscles are pulled. And what that does is it makes sure that blood can't flow backwards, that the valves stay patent, etc. Whereas when the atria undergo diastole, or the heart undergoes diastole, then those muscles relax and the valve closes and prevents retrograde flow of blood. So the function of the atrioventricular is fairly simple. Semilunar valves operate in quite a different way. These again have sort of cusp structures, but they're almost like little sacs. And when pressure becomes greater in the ventricles than in those great vessels, then blood is forced up through the valve. And that opens up those cusps. As the pressure then begins to fall in the ventricle and it starts to get a little bit greater in the vessels, then blood begins to sort of move backwards. And as it does so, it falls down into those cusps, fills them up, and that pressure closes them. So that's what prevents blood from falling backwards. As it falls backwards, it shuts the valve, and then it can't get back into the ventricle. So the semilunar valves work quite differently. This is particularly important, really, in regards to the aortic valve, because either side of the base of the aorta, you remember you've got the aortic sinuses, which provide the outflow passage to the coronary arteries. And that backflow of blood into the cusps allows those sinuses to then fill up with blood and release it to the coronary arteries. Which is why you try and prolong diastole in patients that have got coronary heart disease. Because once the ventricles relax and the pressure becomes greater in the vessels, then blood's going to fill up in those cusps and perfuse the coronary arteries. So that's really, really important in coronary artery perfusion. Again, just a diagram to show you where the pulmonic valve is, at the origin there, the pulmonic arch, and then we have the aortic valve, which is here. And sometimes diagrams don't bring this across fairly well, but the orientation of these is more or less exactly as you find them in the body. You can listen to these valves, and you listen to them in particular points in order to amplify the sound of a particular heart valve. So you can see these various different letters show you where the different valves are. So A is showing you where the aortic valve is best heard, which is sort of halfway down the body of the sternum. And then just up from that we have or superior from that we have the pulmonic valve, and then we have the tricuspid valve here, and mitral valve, which is more or less corresponding to the apex of the heart. So these positions we'll go through a little bit more later on in this series, but these are essentially the introduction to cardiac auscultation. First heart sound, closing of the AV valves. Second heart sound, the closing of the semilunar valves. The valves can become diseased, and this is an example of a heart valve where you can see these are the chordae tendineae, and this is all streptococcal infection. This is Streptococcus pyogenes that's caused this. It's an example of a patient that had rheumatic fever. They've developed all these little tiny blood clots on the surface here, and it's very badly infected. Now, not surprising, this patient died because we're looking at a post-mortem image, and what you can see is very red, inflamed areas. This is very angry looking tissue, and so this tells us that this patient probably had quite nasty endocarditis, which may have been caused by rheumatic fever, which is a disease where you will get antibodies attacking the surface of the valves. But this is an endocarditis, an infection caused by Streptococcus pyogenes. So that's what can happen if the valves become infected, and they'll lose their function. So they get eaten away, and often the only way to sort that out is to put a valve replacement in. So finally then, let's look at the anatomy of the pericardium. Now the pericardium is split into two layers. It's split into a outer fibrous layer, which is the hard tissue covering. This is richly innervated. It's innervated by the phrenic nerve, and it receives blood supply from the pericardiacophrenic artery, which is a branch of the internal thoracic artery, and then also pericardial branches of very different arteries in the thorax and the abdomen. So you get some that come directly off of the thoracic aorta, you get some that come off of the bronchial and esophageal aorta, arteries, etc. So there's quite a few pericardial branches of surrounding arteries that will supply the pericardium. And then we have the serous pericardium, which is again split into two. So we have the visceral serous pericardium, which is adherent to the myocardium, and then the parietal serous pericardium, which is adherent to that outer fibrous pericardium. We've shown that on this diagram here, so that dark blue zone there would be the fibrous, and the text has come over a little bit, but this should be the parietal visceral. The space in between them is known as the pericardial space. Now that should be very small. It's amplified here to demonstrate the point. But it's what we call in anatomy a potential space, which means that it shouldn't really be there. There should be just a thin, lubricating layer of fluid, what we call pericardial fluid. Shouldn't be very much of it. It should literally be very, very, very thin. And this is to assist in the movements of the heart as it contracts, to make them fluid. Now, you can get various different diseases that affect the pericardium, one of which is pericarditis. So the layers of pericardium can become infected, and this characteristically produces a rubbing sound when you listen under the stethoscope, so you can hear sort of a rubbing. It's quite difficult to describe, but very characteristic when you hear it. And when people do have pericarditis, they often complain of a chest pain, which might be suspicious of an acute coronary event. But one of the ways you can distinguish it is asking the patient to lean forward. And if they say that that pain is relieved when they lean forward, then you might be looking at pain from pericarditis. So pericardital pain is often relieved when leaning forward, and that's a distinguishing feature. Now, it can also be affected by something a little bit more serious, but we'll talk about that shortly. This is a dissection of the pericardium, and we can see the pericardium has been reflected, which means that we've folded it away. And you can see that superiorly the pericardium is continuous with the roots of the great vessels, which would be the pulmonary arch, the aorta, etc. And then inferiorly it attaches to the structure down here, which would be the diaphragm. It attaches to the central trefoil tendon of the diaphragm, so it's going to be adherent to that zone there. And then either side it would be continuous with the pleural cavities, the pleura, which is known as the mediastinal pleura. So either side it's continuous with the mediastinal pleura. Superiorly the great vessels, inferiorly the central trefoil tendon of the diaphragm. The anterior portion of the pericardium, which we can't see, it's been reflected here, contains quite a thick ligament that's known as the sternopericardial ligament that attaches the pericardium to the body of the sternum. There are separations between the visceral and parietal pericardium, which are known as the pericardial sinuses. The transverse sinus is one that you hear of, and this sits as a space where you could literally run your finger in front of the superior vena cava and behind the aorta and pulmonary artery, and that's known as the transverse sinus. The other one, which is more difficult to visualize here, is an oblique sinus, what we call the oblique pericardial sinus. It's a J-shaped sinus, and it is literally that space that you can see demarked by the arrow there. It's a space between the chambers and the inferior vena cava, etc. It's quite difficult to visualize on a 2D image, but if you could literally put your hand underneath the heart right to the back, then that would make a J shape, which is the oblique pericardial sinus. This is important because important in surgery where we've got a space to be able to access the vessels. So to say there's spaces where the parietal and visceral layers part, and it allows easy surgical access. This is not a normal chest radiograph. This patient looks like they wouldn't be very well. The reason for that is because we can see a very large heart shadow. Now, granted, this has been taken in an AP field, but look at this here. Can you see that shadow? Shows a little bit of what we call tracheal deviation. We can see they've got leads going all around here. So this patient isn't very well. What's happened then? Why is that cardiac silhouette so large? Well, they've suffered from what's called a pericardial effusion. And that pericardial cavity or pericardial space that we talked about before should be very small. But you can get fluid leaking into that cavity, increasing pressure and putting pressure down on the heart underneath it. And that's exactly what's happened here. Now what this is a transverse slice via CT imaging. And what we're looking at is, as you can see, this is the heart. This is the aorta. We're looking up from the patient's feet at this point. Okay, so we're looking up from the patient's feet. So this will be the thoracic aorta, this large structure here. And all of this is blood. This is the heart. All of this is blood. And what has happened is this patient has bled into the pericardial space, and that has a posh name called cardiac tamponade. What happens is blood collects in that space and it begins to place pressure on the heart. And you'll know that Starling's law of the heart states that ventricular filling determines the cardiac output. If you have pressure placed on the heart, the ventricle can't expand, it can't fill as much during diastole, and what happens as a result is the cardiac output is compromised. And the classic presentation for cardiac tamponade would be a patient that has arterial hypotension and a raised venous pressure, and then displays what we've just seen previously, which is a large cardiac silhouette. And this is quite dangerous. This can kill people. It is associated with trauma to the heart, so if you stab someone in the heart, obviously blood's going to leak out and fill up into that pericardial cavity, but it's also associated with something else called aortic dissection. I've seen a case before on post-mortem where a aortic dissection resulted in bleeding into the pericardial cavity. At the base of the aorta there was a tear, blood leaked out into the pericardial cavity, and it can cause sudden collapse and death if it builds up that rapidly. So it is something to look out for. So that covers the end of that particular tutorial. You can download the notes online, and you can also take part in the quiz to test yourself on the understanding of this topic. Thanks very much for listening. Don't forget to visit the website www.theanatomyresource.co.uk.